going through this semester lah. Alright, so this is the expected time where we will do or conduct our laboratory session, right? Which means that we may start by next week lah, on genetic transformation of tobaccos, and then we will try to do some mitosis experiments. Alright, and the yellow one will be conducted by me, the white one will be conducted by Dr. Zarina lah. So currently we received the news that on week 3 we can conduct it face to face. However, please wait until the confirmation by the course coordinator lah, ataupun the lecturers yang involved lah later, sebab it's in plan we can conduct a face to face laboratory session. However, if anything new information comes during that particular time, so we may change it. We might change. I'm not too sure. Tapi as for now, we will stick with the plan that by week 3 we can conduct any FYP or laboratory that needs some practical to face lah. Alright. So this one assignments. Alright, it will be one by me, one by Dr. Zarina. I will conduct the topic and give it to you guys later. Same goes with the presentation as well. Alright. So bear in mind there will be presentation and assignment given by me and Dr. Zarina. Alright. So this is the midterm. It will be conducted by me. Alright. 20% as we talked last week. Alright. CLO1 and CLO2. And Dr. Zarina will conduct the final examination. CLO1, 2 and 3. Alright. So dekat sini you guys can see lah kita punya course outline. I will share the course outline in the iTalim as well lah, for you guys for your references as well. Alright, so this is the summary what we and Dr. Zarina discussed since last week before the semester start and last week as well lah. But kita nak tahu how well is the laboratory session can be conducted later lah. Alright, so we already ordered a few material for kita punya examination, kita punya laboratory session as well. Alright, so everyone is ready. Can we start our session today?
Yes.
Alright, so before we go further, let's start our session with Ummul Kitab Al-Fatihah. Oh, by the way, can I ask something? You guys bila sampai hari tu, you guys kena quarantine ke apa and then you guys need to move back to different mahalah. Am I correct? Macam tu
Tak.
Tak. Terus masuk ke dalam mahalah lah. Alright. Tak ada quarantine and so on. Alright. Understand. Alright. So how is in mahalah? You guys boleh keluar ke? Boleh keluar?
Boleh.
Boleh.
I see. That's good. You guys can do your daily routine like usual lah. Boleh pergi library and so on, kan?
Boleh.
Cuma kelas je lah depend dengan kuliah. Am I correct? Kawan-kawan korang kuliah lain, okay. Dah start face to face ke?
Dah hybrid.
Oh, hybrid jugalah. Alright. So, alright. So, hopefully everyone is good. Do take care lah. Do take care as well. Alright. Sebab nowadays, my wife ada klinik. Bila dia buka klinik tu nowadays, orang yang datang kebanyakannya kalau demam dan yang ada simptom tu mostly positive. But don't worry too much. I think it's quite common nowadays, right? So just do take care of yourself. I mean, do self isolation and so on. To be honest with you guys, I was also CC1 since I also CC1 last time in last 2 weeks, I guess. Sebab mother dia positif, so dia duduk dengan saya lah. Tapi dia duduk quarantine kat rumah, but I was the one yang jumpa dia. So I just need my self isolation and so on. But do take care lah. But don't worry much about that. I believe you guys already have taken your second dose and also booster as well. Alright, those yang belum lagi just do you can do so. And any places available, you just can come and just do your booster lah. Right, so hopefully everything is good. Alright, so everyone is good today. Oh, by the way, nak tanya, maybe this is second week lah. Everyone ada kelas ke pagi daripada 8 sampai 10? I mean any other subjects ataupun you mesti required courses and so on. Ada tak?
Hari ni tak ada.
Hari ni tak ada. So maknanya hari lain adalah
Ada yang ada.
I see. Alright. So maknanya we stick with 10 lah kot, I guess, Mas. Sebab so I believe next week kita start dengan pukul 10 lah, macam usual lah. Alright. So I don't want to make such hassle for everyone. Takut ada kelas dah terkejar-kejar nak masuk kelas and so on. Alright. So maybe we can stick with 10 lah, Mas. Next week. Starting next week lah, insyaAllah. Any changes, I will let you guys know. But if no changes, we stick with 10 lah, insyaAllah. Alright. So for today's continue. Yes, saya. Record
Bukan, Sarah tak boleh masuk lagi.
Oh iya ke? Kenapa
Tak tahu, dia kata dia tak boleh masuk lagi.
Dia tak boleh klik I think there's one of your friend ada contact saya yang dia kena move. Oh ni Sarah? Alright. Dah masuk dah? Hai Sarah. Boleh masuk? Boleh masuk Hai Sarah. Ada
Ya, boleh.
Alright. So by the way, I come to realize that we cannot do the we cannot do the record sebab I think Google already upgrade dia punya sistem. And I come to my concern last week bila kita buat VLAC screen recording tu, dia tak ada suara. Alright. So let me try to figure out more ways to do the recording by next week. Maybe I will try to use a Zoom maybe so we can record the session gitu insyaAllah. But for today, is it okay kalau saya tak boleh nak buat record? Or you guys want me to do some record as well tapi tak ada suara? Okay, let's do this way lah. Alright, by next week I will try to use a different platform lah. I guess I will try to use Zoom and we do the screen record by next week lah. I try to subscribe the Zoom lah by next week so we can do the screen recording as well. Alright. Well, I think it's only 10 minutes faster. Alright, apologies on that matters. Alright. So everyone is good. Can we start the session?
Yes.
Alright. So just don't worry. This is more on refreshment sahaja. I believe kalau record pun not really ni lah, but it's just more on refreshment. I believe you guys already know about this matters for today's, but maybe starting next week a little bit different. So that is where we need to do the screen record, I guess. Alright. So this will be our overview of genetics lah. So this is very general. Since last week we learned about the laws, the Mendelian laws. So I hope and I believe everyone can understand. Tak perlu memorize. Try to understand what is actually happened by the Mendel punya study lah. Alright. So how can become 3-to-1 ratio, how it can comes to 9-3-3-1 ratio and so on. Alright. So in research sebenarnya the homologous recessive tu adalah quite crucial sebab it's very rare, susah nak jumpa. Alright. So whenever we get that, then we will try to culture them as much as we could lah. Alright. So that's why I believe you guys know about certain situation of the inheritance lah. So certain phenotypes that you guys can observe is quite interesting. Okay. So related to that, alright, kita akan ada juga laboratory session related to Mendelian punya theory. Alright. So that is where we will conduct at this particular week lah. This week. Drosophila part 1 dengan Drosophila part 2 ni. Alright. So later on 28th of April dan 12 of May. This is actually one laboratory lah sebenarnya, but this interrelated part 1 dan part 2 lah. Alright, so we will do and conduct that insyaAllah later. Kita akan main dengan lalat lah. Alright, so this is a summary figure for today's lectures lah. Alright, so it's coming from the nucleus. Alright, so kita nak tengok the chromosome, what is actually the chromosome, and actually what is the building block of the chromosome. Alright, so this is where it coming to this particular part, the DNA. Alright, they bind from the double helix, they will fall in the histone proteins, and further with the histone protein they will further folding, alright, to become nucleosomes, and then this particular nucleosome will further tangle around and make up the chromosome, right? So to be as a refresher, you guys, I believe you guys already know, right? We have 46 sets of chromosome, right? So 23 pairs of chromosome to be exact, right? The chromosome number 23 is our sex chromosome, which is our gender lah, right? DNA is the building block of life, actually. Right? So this is nucleic acid, right? This is RNA, and then this is DNA, all right? You guys realize what is the differences between both of them? Alright, nampak beza ke? So mostly dia tak ada nampak perbezaan sangat lah ya, RNA dengan DNA, apart from the OH group, right, on the carbon number 2. Right, to be exact, RNA is similar to DNA, but they have different function actually. Alright, so but the main differences between them is the OH group, right, on the carbon number 2. Okay, so nucleic acid. Nucleic acid has 3 distinct chemical parts, right? Contain phosphate group, right? And then 5 carbon sugar, and then a ring-like molecule we call base, right? This is where it will become a little bit different compared to each other lah, right? Yang lain semua sama, phosphate group dengan 5 ring carbon sugar, but this one will be different. Alright, this is where it will become purine and pyrimidine dekat sini, depending on the base pairing. Right, so this is purine and pyrimidine. So how to understand and how to memorize is quite easy lah. Pyrimidine nama dia panjang tapi dia structure je just one lah. Purine nama dia pendek sikit but the structure is 2 carbon structures. Alright, so 5-sided ring pyrimidine. Alright, a 5-sided ring joined with 6-sided ring. This is purine. Alright, just remember adenine and guanine is purine. Alright, cytosine and thymine is pyrimidine. Right, and in RNA it changed to uracil lah. Thymine ni change to uracil. Alright, so how to memorize is not memorize, how to understand, memorize lah senang cerita. Alright, pyrimidine is 1 single structure, purine is 2 structures, and purine is A and G. Alright, this is CT sebenarnya. Pyrimidine adalah CT. Cytosine and thymine. Alright, it's more refresh for you guys lah sebenarnya. Alright, so this is with this different type of base will lead to different type of gene expression later. Alright, kita akan tengok lah what happen sebenarnya with a series of purine and pyrimidine sorting up among each other. Right, this is where it could be transcribed into one meaningful messenger, and this particular messenger will be further translated into protein, and that protein will be the building block of organism, or the protein can become some machineries that can show some function to the organisms. Right, so we are now in the most basic building block, right, of the life. Alright, so differences between nucleotide and nucleoside. Alright, nucleotide dia ada all of these things, but nucleoside they are non with the sugar, with the phosphate group. Alright, so remember this. Alright, so this remember this. Alright, sebab nucleotide ni dia akan bercantum with another nucleotide dekat atas, so they will become the DNA with the double helix punya formation. But if nucleoside is just a single nucleoside structure, right, they will not bind to each other, right, they will just form by themselves. Okay, so remember this particular structure. Nucleoside actually adalah nucleotide actually adalah nucleoside plus the phosphate group. They become the nucleotide. This one, right? So remember this phosphate group, right? I think you guys still remember this particular structure, right? So this particular structure will bind with the one incoming structure, right? One incoming structure of this particular nucleotide, and dia akan bergabung dan bergabung. They will form one particular structure of DNA. This is the thing that I did mention just now lah. Right? So remember this phosphate group, dia akan bind 5 carbon sugar, dia akan bind to third carbon sugar dekat the opposite side. Kat sini. Nampak Ni 5, 5, ni adalah yang ketiga kat sini. Alright, ni ada 5 dan ni adalah yang ketiga kat sini. Dia akan sambung. Alright, so same goes with the opposite side lah. The opposite side ni pula dia akan invert. Alright, compared to this one particular side. Dia akan terbalik lah. So which mean that this will be 3 and this will be 5 carbon. Alright, so this is where it's coming from 5' to 3' or 3' to the 5'. About the sequence of the DNA. Right, so double helix structure formed by this particular backbone. Alright, sugar phosphate backbone. Right, the best pair yang saya cakap tadi adalah purine will always pair with pyrimidine. Well, pyrimidine will always pair with purine lah. So remember, pyrimidine ada apa? CT, cytosine dan thymine. Alright, so purine ada apa? Adenine dan guanine. So adenine, this is the adenine. Right, this is the guanine kat sini. Alright, so A will always will be paired with T, and G will always pair with C. Alright, from this particular structure, this is very important kalau boleh tengok kat sini. Alright, so AT punya bonding, they have 2 double helix, 2 hydrogen bond sahaja. But best pair between G dan C, dia ada 3 hydrogen bond. Alright, so in study dan research sebenarnya, alright, study and research, alright, GC best pairing ataupun kalau kita ada sequence of DNA yang ada banyak GC punya sequence, alright, GC punya sequence, alright, so this actually is very hard to kita nak pecahkan dia ataupun kita nak potong dia lah in a way lah. So kita nak open up the DNA, alright. If we have a lot of GC content, alright, we may need to have a high temperature, alright, atau high suhu lah untuk kita breakkan the hydrogen bond. So that's why when you do some study on DNA, the first thing that you guys may need to observe adalah dia punya base pairing dekat dalam DNA. Oh, siapa yang terkejut tu? Alright. So understand about this particular sequence. Alright. So sebenarnya this particular base pairing ni play important roles of the DNA. Alright. There might be you might be encounter a lot of DNA sequence. Alright. When you see a lot of GC content. Alright. A lot of GC content in one particular sequence. Alright. So this is where you guys may need to think on how to break the bond. But kita nak bukakan dia. So we can do the transcription, right, of that particular sequence. Macam tu lah. Alright. So this is where we can do one step at a time lah. So we learn about this. So nanti saya akan explain why this is important lah. Kenapa ada three hydrogen bond dengan 2 hydrogen bond ni. We will explain later when you guys do some of your laboratory experiment later. Alright. So this is where kita akan pernah dengar pasal PCR ke? Polymerase chain reaction. Have you guys heard about PCR? Hello? Hello?
Hello.
Hello. Hi. Pernah dengar pasal PCR ke? Hello?
Yang test tu ke?
PCR. Betul lah PCR test untuk ni lah. Untuk COVID pun sama juga. Actually dia macam ni. Alright. Let me tell you one concept of the PCR lah. Okay. Simple PCR punya concept. Alright. Since we are talking about this. We are talking about the DNA sequence. Let's talk about the PCR. Alright, PCR ni, alright, dia macam ni. Imagine the COVID-19 punya apa nama? COVID-19 virus COVID-19 ni dia ada protein kat dalam Dia ada protein. Alright, the protein will be encoded by a DNA sequence. Alright, so kita macam mana kita nak tahu the COVID-19 is exist? Alright, so by doing the PCR, by doing the RTK is one thing lah. Kita swab, kita tengok by using the kit, ya, kita akan nampak how the antibody ataupun antigen akan nampak. Alright, another thing adalah kenapa kita buat PCR? Alright, when we talk about PCR, PCR can detect even the slightest existence dekat dalam badan. Alright, kalau ada sikit pun dia akan detect. Alright, apatah lagi kalau banyak lah. Alright, so kenapa kita nak confirm with PCR later? Sebab kita nak mengesahkan, let's say kalau ada even ada sikit pun dalam badan, so which mean that the COVID, the virus is still there, exist in our body lah. Alright, until kalau kita dapat negative result, which mean that kita dah swab, kita tengok dan dia tak ada. Alright, so even that is why people are prone to do the PCR as a confirmation untuk visit dekat mana tempat ataupun nak masuk ke satu tempat lah. So what happen actually in the PCR? PCR ni kalau kita buat swab dan dia akan larutkan dalam solution lah in the buffer. Alright, that is our DNA content lah. Apa-apa DNA pun boleh pakai. Tapi kenapa kita swab dekat dengan dekat rongga hidung tu? Sebab that particular virus living there. Dia duduk dekat dalam tu. Alright, so what happen bila kita swab dan kita akan letak dalam buffer. So what happen, dia akan dihantar kat dalam lab lah. So if you ask me, I can do that as well. Tapi not certified to do so sebab this is more on medical legal punya study, medical legal punya eksperimen lah. Alright. So if you ask me to do so, I can do because the concept is the same if ada PCR as well. Alright. Tapi with the extra precautions lah. Alright. Orang pakai PPE, handle with proper medical punya apparatus. Alright. So dia akan swab, dia akan ambil dalam buffer tu ada DNA. What happen? They will do the PCR thing. Alright, what happen in PCR? Alright, PCR ni in short ni adalah they can amplify. Dia boleh banyakkan DNA yang ada dekat dalam tu even though dia sikit. Alright, dia boleh banyakkan sebab kita bila saya cakap ni even the slightest amount yang ada dekat dalam badan dia boleh detect. Alright, apatah lagi kalau banyak lah. Kalau banyak dia akan detect lagi banyak lah. Alright, so the function of PCR ni adalah with one particular sequence yang sikit, dia akan amplify dengan banyak. Alright, dia akan amplify dengan banyak because one cycle of PCR dia can amplify double, almost double. Lagi banyak, dia lagi banyak the amount, lagi banyaklah dia akan amplify. Alright, so this is where they will amplify that particular DNA sequence yang ada dekat dalam dan dia nak check ada ke tak ada. Alright, so how they amplify? Dia menggunakan primer. Alright, so primer ni a short DNA sequence lah. For you guys, don't worry, jangan rasa pening dan risau. I can explain it later sebab memang you guys will may need to learn about it later. Alright, so they're using a very small DNA sequence yang ada dekat depan dan dekat belakang. So which mean that apa-apa yang di tengah-tengah tu dia akan amplify. So normally apa yang kita akan amplify adalah protein yang coming from the COVID-19 virus lah. Alright, in between them. So kita ada dekat hujung dan dekat belakang. So kita apa-apa dia akan bercantum together, dia akan ambil lah. So let's say sequence daripada sequence 1 sampai sequence 10. Alright, sequence yang short DNA sequence tu ada dekat nombor 1 dan short DNA sequence lagi satu akan berada dekat nombor 10. So yang satu tu dia akan pergi ke sampai nombor 10, yang nombor 10 dia akan pergi ke sampai nombor 1. So this is the 5 prime, the 3 prime dengan 3 tadi yang terbalik ni. Right, this one yang terbalik. So dia akan amplify anything in between that. So apa yang ada dia akan keluar. For example, you get apa yang dia try to amplify is not there. So itulah pengesahannya adalah so you get the negative result of the PCR. Alright. So if positive, they will amplify inside. Apatah lagi kalau sikit, kita boleh detect lah. Apatah lagi kalau banyak lah. Alright, so this is the little bit concept of PCR. They are using this particular concept, understanding of the DNA sequence. Alright, so dia akan ambil a little bit of DNA sequence dan dia akan amplify it to check. Alright, so this is double helix that I did mention just now lah. Alright, the triple bond dengan the double bond of hydrogen kat sini. Yeah. Alright, so lagi banyak this particular base pair, lagi tinggilah temperature dia. Alright, kenapa saya cakap temperature tinggi ni? But when you do the PCR, alright, when you do the PCR just now, you need to break ni. Kita kena potong DNA ni, dia jadi single stranded. But now dia ada double stranded, betul tak? Alright, dia ada double stranded. When we do the PCR, kita kena potong ni dan kita akan pairkan dia with kita punya buffer. Ataupun kita cakap DNTP kita lah. Ini adalah dia punya base pairing ya, complementary base pairing between adenine dan guanine. Tapi bila kita dah breakkan them, we break them here, right, we put our detection kit kat sini. Alright, DNTP kita. So dekat situlah dia akan amplify jadi banyak. Kita letak yang kita akan letak DNTP, contohnya A dan T, betul tak? Alright, so kita akan letak T yang kita buat akan bind dekat sini. So dia akan jadi banyak. So T ni pula dia akan bind dengan A yang kita provide. So dia akan jadi banyak dan banyak. Alright, so this is concept of PCR. Alright, so ini parallel dan anti-parallel lah. Macam saya cakap, this is from 5' to 3'. 5, 3, 5, 3, 5, 3. Alright, so this is 3, 5, 3, 5, 3, 5, 3, 5. So alright, dia anti-parallel. Alright, so this is where the 5' to 3' is coming from lah. Alright, so this is the actual structures yang kita boleh nampak. Alright, this is 5' and so this is 3'. So this is y the direction. 5 to 3 direction. 5 ke 3 punya direction. Alright, so in between adalah base pairing of the base. Alright, adenine with thymine, right, AT, alright, thymine with A, adenine, T dan A, alright, then cytosine with guanine. Alright, so this is the triple bond, this is the double bond. Right, so this is I did mention just now, 5' to 3' to 5', right? 3' to 5' kat sini. Dia akan bersambung. Dia akan jadi anti-parallel. Right? So this is the double helix structure. Alright? So I hope you guys can understand this figure so you can easily understand how things work in the real world. Sebab I know this is a little bit hard to figures dia banyak, konsep dia banyak, but understand this one in the schematic diagram punya condition, maybe will be easier for you to understand. And when you look at this, you have the basic ideas what is in mind. Right? So adenine dan thymine ni, this is the double bond that I did mention just now. Right? Adenine ni adalah daripada purine, this is daripada pyrimidine. Why I say this is purine? Because there are two ring structures, 5 carbon dengan 6 carbon. Right? This is purine, this is pyrimidine, only one ring carbon structure sahaja. Alright? So this is where the consists. Nampak tak dekat sini? Alright, so this is double bond to triple bond. Alright, so this will be the concept. Alright, any questions before we go further? Any soalan?
No.
Oh, alright, so just more refreshment kan? So I believe you guys already understand about it. So now let's talk about chromosome. Alright, to be honest, when I was a student, I am confused. What is DNA? What is chromosome? Alright, so where is the chromosome located and where is the DNA is located? Alright, actually DNA is what built the chromosome, and chromosome ni dia akan form when the cell is about to do the cell division. Kalau dia tak buat cell division, they will be in the non-folded form, where they are free structures, alright, as a part of the DNA. Dalam bentuk DNA lah, dalam bentuk this particular form. Alright, so when they are about to do the cell division, so this is where they will pack everything, alright, they will pack everything in this particular form. Just imagine macam bila awak nak berpindah, sebab tu saya tanya awak tadi, awak sampai macam mana and so on. Alright, kalau awak dah sampai, awak akan kalau awak sampai kat satu tempat, awak bawa kotak dan beg lah kan. So awak akan letak dan susun dia dekat mana location dalam bilik awak. So when you about to move out, alright, what happen? You pack everything in folded form, masuk dalam kotak ke, masuk dalam bag ke, and then you move out to other place. So this is where the concept is a little bit similar macam tu. Cuma when they are in this particular folded form, what happen adalah they make a copy of this. They buat another copy of them in the form of chromosome, where they will share this all the information to the next cell yang akan dibuat lah. So they will divide. So all the information will be the same in the new cell. Alright. So then after the cell division, they will disperse back, move out everything, and they will be in this particular DNA form. Right? All scattered. Okay. So this is the chromosome I did mention just now. A long strand of genetic information located in the cell nucleus. Right? Formation double helix DNA wrap around protein called histone, which I did mention just now. And then to form the nucleosome, right? So di dalam bentuk histone dia akan bind with the histone protein. So protein dalam bentuk bulat ni lah. So dia akan bind with the histone protein, alright? The condensed structures. And then when they bind with the histone protein, they will further bind themselves, right? To become a nucleosome, alright? So this will be the chromatin punya formation lah, alright? Chromatin is just one single structure, right? Then when they have this particular full folded structure with two arms, all right, two parts and with two parts and two arms, all right, so they will become the chromosome. This is arm, longer arm, and this is shorter arm, and they have the centromere dekat tengah, right? This is chromatid, one single structure, right? So this is centromere. The function of centromere adalah to position them in the equatorial form. Then this is where, right, centromere is where the microtubules will bind to them. Right, dia akan pecahkan structure dia later lah. I believe everyone understand about the mitosis formation. Right, so this centromere plays important roles during the mitosis punya process lah, with this the cell division. Right, tadi mention that nucleosome, and nucleosome coils and form the chromatin. Right, this is the chromatin. Chromatin wrap around each other and from chromosome. This is where it's formed. Alright. So what happened? Chromosome has two arms, short arms and longer arm, P and Q. Centromere holding the chromosome together at the center, and half pair of the chromosome is called the chromatid. Alright. So this is where actually the chromosome are formed. Right. I did mention just now. Like the mitosis punya experiment, alright, yang macam kita ada salah satu mitosis punya eksperimen tu, where we want to observe the chromosome punya structure by doing it in the onion punya sel bawang lah. So kita akan ambil sel tu dekat, kita akan ambil sel bawang dekat akar lah, right? So akar yang tengah divide. Dekat situ kita akan tengok the formation of the particular structure. Kita boleh nampak. I believe you guys already knows about process of mitosis. Alright. So don't worry, we will talk about that in chapter 4 later on how the process one by one. Alright. So any questions before we go further? Soalan? Ada yang terbuku di hati ke? Tak ada. Alright. Can we proceed? Kita nak break for 5 minutes. Proceed ya? Alright, kita proceed. Don't worry, this is a very leisure punya talk ya. Alright. So remember this, alright? This is the concept yang kita awak kena faham. Dan everyone kena faham lah. Alright, transcription dan gene expression. What actually happened kat sini. Right? So this is the central dogma of molecular biology, right? Central dogma of molecular biology, which is that DNA makes RNA and makes protein. DNA tadi yang kita belajar, they will make the mRNA, and this mRNA will create a protein. Right? This concept that you guys need to understand. Alright? So whatever DNA yang ada, right? Macam saya cakap tadi, when you do the PCR, DNA of the virus is there, alright? So we are extracting them, and this particular sequence yang kita extract tu ataupun kita amplify tu tadi is a form of protein yang akan dibuat oleh that particular virus. So this is where we can have this particular understanding. Alright, DNA will undergo transcription where they will form the mRNA punya form. Alright, so mRNA will translate to become a protein. I believe everyone already know and understand the process of transcription dengan translation process. Let me assist you to just refresh your understanding lah. Alright, so what happen dekat DNA? Right, DNA ni akan dipecah, double helix ni akan dipotong. They will be transcribed. Anything yang akan dalam ni akan di-transcribe to form mRNA. Right? If you guys realize dekat sini double stranded and mRNA is formed as a single stranded form. Right? When you talk about mRNA, mRNA always in the single stranded form sahaja. Right? So this particular single stranded form will be translated by a process where they will form a protein. Nah, ceritanya adalah awak ada buku dekat library, awak copykan dia, and copy tu awak dapat, and bila awak dapat copy tu, awak try untuk buat copy tu. Contohnya awak dapat resepi lah, ni buku resepi. Awak copy buku resepi tu dalam sehelai kertas, and di sehelai kertas awak akan buat menjadi kek awak yang nak buat lah, ataupun masakan yang awak nak buat. But this is the concept, process ni. Alright, awak tak boleh bawa semua DNA ni untuk jadikan protein, tak boleh, sebab the DNA is fixed inside the nucleus. Alright, only information from the DNA can be carried out and be copied lah. Alright, so this is what happens. Alright, dalam nucleus kita ada DNA, and DNA ni akan di-transcribe. Alright, when it being transcribed, dia akan dalam bentuk RNA punya form. So dalam bentuk RNA ni pula dia ada intron dengan exon, where I believe you guys have heard about it, right? So kita nak remove all the introns, the unwanted information. So we just keep all the exons sahaja. So this is what we call RNA splicing. Kita removekan dia. So what happen kat sini adalah only the exons sahaja yang ada. So this particular mRNA, right, di dalam nukleus ni tadi akan dihantar keluar ke dalam cytoplasm of cell. Bila dia hantar kat luar, what happen? Dia akan disambut oleh ribosom. Ribosom ni lah yang akan menyambut dia bila dia keluar. What happen? Ribosom ni adalah kilang yang akan translatekan this messenger kepada protein, which is the translation process. Alright? So this protein akan dihantar keluar through the Golgi apparatus. Okay, sel ni, protein ni pula akan bergerak ke any location inside the organism that requires them. Alright, contohnya lah untuk surut bengkak, protein yang untuk surut bengkak dia akan keluar pergi kat tempat yang ada wound tadi, dia akan pergi kat situ, dia akan surutkan bengkak. Like that. Kalau suhu temperature tinggi, what happen? Another protein akan pergi kat situ to trigger atau to acclimatize the temperature dekat area, certain areas for them, like heat shock protein and so on. Alright, so this is the concept. Alright, so the protein synthesis, the human genome contains around 30,000 genes. Each one of them encode for one protein. Alright, DNA holds the code for protein, but it cannot pass through the nucleus. Alright, protein synthesis is performed by ribosome in the cytoplasm, and cell use RNA to copy the code from the DNA and bring it to the ribosome. Konsep yang saya cakap tadi lah. Alright, DNA ni tak boleh keluar, they need to be transcribed, which means you make a copy of them in terms of the mRNA. And what happen, this mRNA can pass through the nucleus, right? Then it will be translated later. Alright? So, macam ni, yes. If DNA is a recipe books, protein course of a meal sebenarnya tadi. Alright? Recipe of all polypeptide are encoded in DNA. mRNA is a copy of the recipe yang kita ambil tadi. And this recipe travel through ribosome for translation into polypeptide protein. Ada. Alright, protein yang ada nanti kita akan gunakan lah for certain function of the organism. Alright, so transcription process is very simple. Dia ada 3 step. Alright, so this is DNA double helix yang kita ada. Alright, DNA double helix yang kita ada. What happen in DNA double helix tu, dia ada satu region, we call it a promoter region. This one. Alright, I don't want to go into deeper of the promoter region, tapi promoter region ni adalah satu sequence, alright, satu sequence that have TA formation, tata box we call it, alright. So dekat situ ada banyak, where when this particular specific sequence ada, so that is where the transcription could start. Alright, tak kisahlah sequence apa-apa ataupun tak kisahlah genes apa yang ada. So normally one particular genes they will have a one promoter region. Where promoter region ni akan digunakan for the transcription process to start. Right? Contohnya let's say you have one DNA sequence yang panjang. Alright? So the one DNA sequence yang panjang tu they will have several promoter region kat dalam tu. Alright? So whenever RNA polymerase, right? RNA polymerase is just another protein, right? That will bind with the promoter and where they will unwind the DNA double helix sequence tu. Dia akan buka sikit untuk sementara waktu. When they open it, they will inform the NTPs ataupun mana-mana NTPs yang available, right? Purine or pyrimidine yang available to bind with the opening of the DNA sequence tadi. Dia akan masuk. Then this is where the RNA sequence will be formed through the RNA polymerase. Right? So remember so initiation form, dia ada 3 step tadi. Initiation, elongation dengan termination. Initiation form ni where RNA polymerase will find a specific protein that they will transcribe to start the transcription process. Alright? Imagine the DNA helix, dia ada banyak sequence of that. So there are a lot of promoter region. So how the RNA polymerase can find the promoter region? This is where another topic that we can talk about in plant biotechnology later lah. Alright? Sebab in order to bind to specific promoter, they will have a trigger. Dia macam ada switch lah. Okay, kita perlukan this particular protein. Tolong pergi dekat promoter untuk transcribe. Dia akan pergi. Something like that lah. Alright. For this particular concept, to study, kita akan start lah. Initiation process, RNA polymerase will bind to a promoter region. Alright. So what happen when they bind to the promoter region? They will unwind the double helix punya structure. What happen dekat sini? Dia akan transcribe all the sequence tadi. Start daripada promoter tu tadi. Dia akan start to transcribe apa yang ada dekat dalam. Right, dia akan pass through all the information, and when they pass through all the information on the single stranded punya structure of the DNA, right, they will form dia punya product which is the RNA. So macam mana RNA ni terbuat? Through the RNA polymerase where they will bind all the NTPs. NTPs lah, NTPs ni adalah nucleotide tadi lah yang saya cakap tadi. Alright, nucleotide yang ada dia akan yang available dekat dalam this particular environment. Dia akan panggil, bind dekat sini, bind dekat sini, bind dekat sini, bind dekat sini. So this is where the structures are forming through the RNA polymerase. Dia akan bergerak from 5' to 3' normally. Right? Dia akan pergi dekat sini. So until dia akan jumpa stop sequence. Right? When they form ataupun jumpa the stop sequence, right? RNA polymerase ni akan release themselves from this particular sequence. Right? Bila akan release, when they release, dia akan cut the RNA yang dah ada, and then this particular double helix akan bind balik together through ligase punya protein. Alright, so this is where the process are formed. Right, so they will bind to the promoter region, they will unwind the double helix DNA, they will form elongation of this particular DNA sequence yang dia dah buka tadi. Alright, so they will have NTPs yang ada available to bind to the complementary sequence yang dia lalu and forming the RNA. Alright, so RNA will be in the form of single stranded sahaja. Right, then when they find the stop sequence, right, RNA polymerase will detach themselves from this particular DNA sequence. With the detachment, they akan release the single-stranded RNA yang dia form tadi. And then the double helix DNA will ligate themselves back together with the ligase punya protein. And then we will have this particular sequence yang dalam the form adalah dia color merah lah. This particular form of RNA we call it as a messenger RNA, mRNA. Alright, so this mRNA yang akan pass through the nucleus punya membrane, then they will go to the ribosome punya. They will go to bind with the ribosome later in the cytoplasm. Right, so what happened dekat sini lah. Right, termination RNA polymerase detach when they reach the stop sequence and release a copy of single-stranded RNA just now. RNA nucleotide different from DNA nucleotide. With the A will pair with U, uracil, which is I did mention just now. A normally bind with T, right, thymine. However, in RNA sequence, right, thymine will be changed to uracil. Right, RNA will undergo treatment. They will be removed the non-coding region, which is the intron. Right, they will just bind by the exon punya formation. Remember this structure. Right, so bila dia dah buat transcription tadi just now, right, they will transcribe everything yang ada dekat DNA. Right, so normally we do not want the non-coding region dekat dalam ni, yang color hijau ni. Right, color hijau ni kita tak nak. So we will remove ataupun splicekan them and remaining only the exon sahaja. Right, this is called the RNA splicing. Actually, it looks simple, but RNA splicing required 7 steps sebenarnya, in detail lah sebenarnya. But for our concept, we do the RNA splicing, remove the introns and recover back all the exons saja, dan we have the complete process RNA yang kita ready to be exported out. Right? So of course you guys may ask, right? So this is 5' to 3'. What happened from 5' here to 3' here? Right? So this is where some study has been conducted. Yeah? Alright, actually some of this also been transcribed actually, tapi dia akan form a patchy RNA fragment. Alright, dia akan 5' to 3' dia akan berhenti, 5' to 3' dia akan berhenti untuk certain period. Alright, so this particular fragment yang form here, alright, will not be used for the mRNA sequence. Alright, but up until now is still be under study. Right, this is what we call as a Okazaki fragment. So Okazaki, which mean that the founder of that particular sequence lah yang ada dekat sini. Right, they found that some of this particular sequence is also being translated as well. Kenapa dia jadi patchy? I said because dekat sini when the RNA fragment, right, RNA polymerase bind to the promoter, dia akan buka dekat sini 5' to 3'. Dia akan follow the flow of 5 to 3 dekat sini. Right? Kenapa di atas ni patchy? Because dia akan unwind sikit demi sedikit. Right? So what happen bila dia bergerak dekat sini, only shorter fragment yang akan dapat 5' to 3'. So when they already transcribe them, dia akan bergerak lagi 5' to 3' balik. Dia akan sikit sampai ujung. But here they are continue from 5' to 3'. Bergerak saja. Sebab dia memang dah dalam bentuk 5/3 dekat sini. Alright. Kalau dekat sini tadi dia 5, dia bergerak sikit baru ada 5 dan dia akan transcribe balik lah ke sini jadi 3. Bergerak lagi baru ada 5 kat depan dan dia transcribe balik ke sini sampai sini. So dia akan jadi patchy lah. So this is also a study being conducted over here as well. And maybe you guys also ask, right, what happened to the introns actually? There are some cases, right, the intron is not really being removed, but they are also important for certain sequence as well. But this is still under study lah as for now, right? Why we have the intron sequence dekat dalam? We call it non-coding sequence lah. Tapi there are some sequence are quite interesting to be studied as well inside here. Alright? There's a lot more opportunity to be done ataupun study in the future, insyaAllah. Right? So this is what happened after the transcription process. Ni yang saya cakap tadi lah. Kita remove all the intron and we just keep all the exons sahaja dekat dalam. We call this one as the RNA processing. Alright, so remember when we talk about RNA processing, there are a few more things that you guys may need to know. Alright, satu, kita buang introns. Kita dah buang intron, kita just remain all the exon. Alright, another thing is that when we say about RNA processing, there are another two things that happen. Alright, they will cap this particular mRNA with C cap, carbon cap dekat depan. And this is where they will endkan dia dengan poly A tail dekat ujung. Alright, there's a lot of A punya sequence dekat ujung ni. Alright, so dia ada carbon sequence dekat depan cap ni untuk tutup dia. So which mean that when they undergo RNA processing, they will remove all introns, they will form a 5 ataupun carbon cap dekat depan, and they will endkan dia dengan poly A tail dekat ujung. So which mean that all the package are secured. Dia ada cover dekat depan dan dia ada cover dekat belakang. Right? If you go into detail about this particular RNA processing process, right? So actually the carbon cap ni is very important for them to pass through the nucleus membrane, and the poly A tail is very important for the binding of them to the ribosome yang akan terima dia. Right? It looks simple but it's more than meets the eyes lah sebenarnya. When we talk about the RNA processing, this is the thing that we talk. Remove the intron, bind the exon back, put the C cap in front, and put the poly A tail kat ujung. Alright, they will be a solid structure where they can pass through the mRNA, the nucleus membrane, right? And where they will be attached by the RNA punya sequence kat dalam. Attached by the ribosome dekat luar. Sorry. Right. So after transcription process tadi, kita dah dapat the matured mRNA. Right. What happened to the matured mRNA? They will be collected by ribosome dekat luar. Ribosome ni lah yang akan translate them into a functional protein. So right. So this is what happened when we do the translation. Translation is a sequence of 3 nucleotides. It's called a codon. Codon satu-satu ni lah. Alright. Tak nampak lagi. This talk about here lah. Alright. So normally when we have a mRNA sequence, alright, of course it looks like a DNA sequence but in a form of copy lah. Alright. So what happened? The sequence DNA dekat dalam tu, before they be translated, we need to understand what sequence yang ada dekat dalam tu. Alright. So that's why we need to understand about this particular codon punya sequence lah. Tak perlu hafal, ya. Yeah? Kita dah ada dia punya table. But the important things adalah you need to know what type of amino acid yang dia akan form kat sini. Alright. So which mean that contohnya lah, contoh alanine. Alanine is the most inert form of amino acid. Dia tak ganggu pun. Kalau berubah pun dia tak akan terganggu mana satu. So in order to form alanine, you can have this particular 4 sequence, right? GCU, GCC, GCA ataupun GCG. All of this sequence dia akan form alanine. Alright? So same goes with adalah serine. UCU, UCC, UCA dan UCG. So this is the serine punya structure. Alright? So in your messenger RNA sequence, alright, kita akan ada this type of sequence yang ada. Banyak. Alright? So the first thing first, what you may need to understand is that the start codon, which is in the AUG. Right? This is one. Methionine. This is in code a methionine punya amino acid. AUG ni dia ada only one sahaja. Dia ada satu sequence saja AUG, which is this is the only start codon yang akan ada. Alright? So whenever you have the messenger RNA just now, alright? When you have the messenger RNA just now, what happen dekat sini messenger RNA, they will find a AUG sequence dulu. Right, AUG sequence akan di-identify dulu dekat dalam ni. Where that is the trigger to start the translation punya proses. Remember tadi kita ada promoter region dekat transcription. Right? So dekat translation pula we need to have one specific region, we call it start codon, where encodes only AUG sequence sahaja. So what happened with the AUG sequence just now? Alright. You can see from this particular schematic diagram lah. Alright. So remember, this is small subunit of ribosome. This is a large subunit of ribosome. What happened adalah messenger RNA dia akan bind first to the small subunit. Right, they bind first to the small subunit. So this is where the mRNA binding site lah. Dia akan bind dulu kat sini. So maknanya mRNA dia akan pass tu panjang kat sini lah. So this is where dia akan attach. The next one is the large subunit akan datang. What happened to the large subunit? The large subunit akan bind according to the P site dulu. P site ni adalah the binding site. This particular P site, dia akan cari AUG site dahulu. Alright, dia akan bind to the AUG site dahulu. What happened when they find the AUG site? They will stop. Alright, so this is where they started the starting of the translation process. P ni will bind to the AUG site dahulu. Okay, what happened when they already bind here? They will build tRNA ataupun RNA yang akan bawa amino acid masuk ke dalam particular ribosome ni. Kan jadi kilang lah. Dia adalah pembawa amino acid ke sini. Alright. So you see from here, alright, the last subunit dia ada three main structure. Dia ada E, exit site. Dia ada P, peptidyl binding site. Dan dia ada A. Alright, this is amino acid binding site. Dan dekat P ni pula dia ada exit tunnel untuk riliskan the binding of the amino acid. Dia akan keluar panjang-panjang kat sini. Alright. What happened dekat sini adalah, you can see from here, right? See from here, yang color merah ni adalah tRNA. tRNA. Jangan confuse with RNA lah. This is tRNA. Transfer RNA kita panggil. tRNA. What happened with tRNA? tRNA ni dia akan membawa amino acid atas kepala dia. Right? Dia akan bawa amino acid dekat dia punya one end. Alright? So, tapi this tRNA is very specific. Because this tRNA, dia ada, one is the top part, dia akan bawa amino acid. The bottom part pula, dia ada sequence. Alright? Dia ada codon sequence that is anti-parallel with the sequence yang ada dekat mRNA tadi. Alright? Contohnya AUG, dia akan jadi opposite of the AUG dekat sini. Alright? So, which mean that they are bringing the contohnya kalau this is the AUG, alright, this is AUG sequence, alright, dia akan jadi GAC, alright, dia akan jadi GAC kat sini, alright, sorry, GAC betul lah, dia akan jadi GAC dekat sini, alright, dia akan bind first dekat P site just now, alright, dia akan bind to P site dekat sini. Alright, so what happen, dia ada methionine dekat one end dia. Alright, dia akan ada anti-parallel coding sequence dekat dia punya other end. So which mean to make them complementary dekat this particular region. Alright, so what happen, tRNA with the methionine, dia akan bind to the P site. Alright, so they can bind to P site. The incoming tRNA with another amino acid sequence that are complementary with the another site dekat bawah ni, alright, dia akan bind here dekat A site, alright, the amino acid peptidyl site. What happen dekat sini adalah they will form a polypeptide chain ataupun bond between the two amino acid dekat kepala. Alright, what happen, they will form the amino acid punya peptide punya bond dekat sini. Right, when they form the bond, right, this particular structure will move one codon at a time, which means that they will move 3 sequence at a time, right, which means that they will shift this particular progression of tRNA, which means that the methionine just now will move to the E site, the incoming tRNA will move to the P site. Alright, so which mean that they are making this particular tRNA yang dah tak ada dah tak ada amino acid will be released dekat sini. right? So you can see from here, the tRNA is being released and they are leaving the poly ataupun the amino acid left dekat sini, where they already form the polypeptide bond with the incoming punya tRNA. Alright, so the process will be shifted to one another up until they found a stop codon. Alright, dia akan continue saja macam ni proses. Alright, the thing that I may need you guys to know what happened dekat sini lah ya. So when they have the amino acid, alright, they will bind here and then they will be released, alright. So they will not have any amino acid left dekat dia. So what happened adalah the empty tRNA, they will be recycled back masuk kat sini. What happened to them? Amino acid, right? They will find another inert amino acid dekat dalam this particular cytoplasm. Alright, what happened? They will be charged. They will be charged by ATP. Dia akan di-charge menggunakan ATP. Alright, ATP, so dia akan dapat energy lah dekat sini. So when they have the energy, they will have a capability to bind the amino acid back. Alright, so what happened to the amino acid? This one will be recycled back kalau diperlukan untuk masuk ke dalam. Alright, so nampak tak ni yang tRNA with the amino acid dekat dia punya top. So this is charged tRNA, alright, yang dah masuk. So what happen? They will be cycle dan cycle dan cycle. Kalau diperlukan lah, kalau tak diperlukan, contoh methionine, dia akan pergi dekat another kilang lain lah. Dia akan pergi kat ribosome lain, dia akan pergi untuk start. So they akan move around, something like that. Alright, so this is what they are happening around. So sooner or later, alright, this amino acid will be form a long polypeptide chain dekat sini. When they found the stop codon, alright, when they found the stop codon, they will release everything dekat sini. Dan this particular amino acid pun akan di-cut, right? And then one by one will be removed, right? Small, large subunit akan terlerai. Small subunit pun akan terlerai. Messenger RNA pun akan terlerai. This is from this particular structure kat sini. This is the thing that I did mention just now lah. Mula-mula dia ada small subunit, dia ada mRNA punya binding site. So dia akan bind the tRNA first, and then this tRNA with the methionine punya sequence, the large subunit akan bindkan dia ke dalam P site. Alright, so what happened to the P site? Alright, so dia akan incoming tRNA akan masuk dekat sini. Alright, so incoming tRNA akan masuk dekat sini, dia akan form the polypeptide bond with the one next to him. Alright, next to them lah tRNA. And then they akan form this particular bond together. Then they will move this particular ribosome one codon at a time. Then this particular codon will be shifted. Right, dekat sini, sorry. Ni start kat sini dan kita sambung kat sini. So when it's here, the new incoming tRNA with amino acid akan bind to the A site. They will form the polypeptide bond. And then this polypeptide bond will move to the new tRNA. And what happened dekat sini, they will move one codon at a time, which leads that this particular tRNA without any amino acid will be shifted to the E site and then being released. And the amino acid with the tRNA with the longer polypeptide amino acid will move to the A site, right? And then the new incoming tRNA with new amino acid will bind to the A site. And then the process goes together. And what happened when the process goes together and with the tRNA that have a longer polypeptide amino acid, when the stop codon bind, here dia akan berhenti. What happened when the stop codon is there? The structure of the complex of the structure will be released, right, one by one, which means that small subunit will be released, large subunit will be released, mRNA will be released, the stop codon also being released, the newly emerged of amino acid will be released as well. So this newly emerged amino acid will be polished, then they will be transferred out to the location where they are needed, right?
So sometimes they are forming complex as well. Ini amino acid. So ribosom ada banyak dekat dalam cytoplasm. So they will form complexes to form certain protein function. Tentu proses dia. Alright. So any questions before we go to our last part? Ada soalan? Or you want to rest for 5 minutes? Ada soalan?
Tak ada.
Ada eh? Kita rehat dulu 1 minit lah ya. Sebab kita dah nak habis dah. Sikit je lagi ni. Then we all.
Boleh.
Ha? Nak continue ke?
Rehat.
Nak rehat ya? Alright. Kalau you guys have any question, do let me know lah. InsyaAllah, I will try to assist. Right. We will continue with the histogenetics. You guys ready? Boleh sambung?
Boleh.
Alright, I think just more on around 5 minutes to go and then we done for today lah, insyaAllah. Alright, so let's continue with cytogenetics lah. This is very simple terms, right? So cytogenetics is two different terms. It's a cyto study of chromosome and other cell components, alright? This is cytology, and genetic is the study of inheritance, alright? The study of structure and properties of chromosome, including the chromosomal behaviors during somatic cell division in mitosis and meiosis. Alright, the chromosomal influence in the phenotype and factor that cause chromosomal changes. Alright, so cytogenetic ni, I tell you one thing, in kita punya department, we have Dr. Razik, right? Dr. Razik, he is a cytogeneticist yang kita ada dekat dalam kita punya department yang kuliah, alright? So, his study is more on understanding the chromosome punya behavior lah, right? So that's why I guess during the mitosis punya process nanti I may invite him to our laboratory session. Let's look at the chromosome together with him later, insyaAllah. Alright, so there's a lot of things yang dia buat lah. So they kaji chromosomal behavior from different type of fruits. Dia tengok dari mana asal dia and so on. We can see from here lah. This is where the cytogenetic punya study is quite interesting and important. That we can see and detect the particular inheritance of this particular chromosome ataupun particular species lah yang ada. Right? So the next one, we talk about crossing over. I believe you guys know about crossing over punya events. Alright? So crossing over punya events mostly happens during the meiosis punya process lah. Alright? You guys still remember meiosis punya process where two chromosomes is overlapping each other, right? So meiosis process is where they will produce the daughter cell. Alright, the daughter cell is there. So the daughter cell will be different compared to each one of them, right? Due to this particular process, we call it crossing over. But crossing over is the reciprocal exchange of chromosome parts at the corresponding position along homologous chromosome by symmetrical breakage and rejoining, right? Dia ada crossing over, they will bind and they will change ataupun the genetic material between them lah, but with the particular similar amount and similar position, right? So you can see from here lah, when you do the crossing over, the exchange of one part of the chromosome is there. Just imagine, just now when you learn how chromosome works, alright? So it's a compress of a lot of DNA sequence, alright? So just imagine one chunk of them is being changed and removed. So actually what will happen with this particular chromosome is that they will have completely ataupun totally portion of different DNA sequence yang akan ada dekat dalam. Alright? The portion of them. Alright? So with this particular event ataupun crossing over punya situation, right? So people tend to do a study about genetic linkage. Alright. So what happened in genetic linkage actually? Alright. So genetic linkage is the study on genes of non-homologous chromosomes assorted independently during meiosis. Alright. They exchange just now, crossing over. What happened? Genes on the same chromosome are said to exhibit linkage and are called link gene. Right. Link gene ni kira dia connecting gene lah. The phenotype characteristic they control are inherit together because they are located on the same chromosome. Alright. So what actually link gene sebenarnya? Alright. Just imagine, just now, alright. Gene A and gene B, alright. A and B referred to as a link because they would likely be inherited together. Alright. So C and D are referred to as link because they would link be inherited together. Right? Kalau kita tengok dekat sini just now, alright. So if one portion of them is being exchanged, alright, being exchanged. So like macam C dan D ni, kalau dia ni terpotong, they will be, alright, they will be transferred to another portion of the chromosome. So when we say about C and D, they refer as linked together because kalau this portion is being removed, transfer, they are also can be detected. Alright, C dengan D, dia akan nampak. So what happened is that A and B are not linked to C and D because they are so far apart. Alright, A dengan B ni adalah not linked because they are very far apart. Just imagine just now C dan D ni di transfer to a different portion. when detect with the chromosome, we can detect C dengan D ni ada bersama-sama. Tapi when you want to try to detect A dan B, dia tak ada dalam this particular new chromosome, which mean that A and B is not linked together with C and D. But C and D is linked together because they are being transferred together, right? So crossing over is likely to occur in space between B and C, thereby separating A and B and C and D from each other. But there are cases jugalah kalau dia terpotong dekat sini B ni sahaja, tapi walaupun dia dekat, tapi when we transfer them, dia tak link sebab they are not transferred together. But this is where with these particular ideas and study of crossing over, people are mapping the chromosome sekarang. Alright, so they are mapping them, so one part to another part, so one portion to another portion. They are studying about genes yang ada dekat dalam chromosome. So genes ni bila dekat sini adakah dia bersama ataupun tak. So they manage to do the genetic mapping. But just remember lah, chromosome ni sangat besar dan banyak. It's not being done by one person lah. They being done by a group of researcher, and one group of researcher maybe study about one portion, another group of researchers study another portion, and so on. They will combine all the data together to make a mapping of one particular organism, something like that, right? So this is where the knowledge, all right? So it's the knowledge from the cytogenetics just now, right? Leads us to the understanding of chromosome punya study, and then especially the crossing over. With the knowledge of crossing over, we manage to do and understand about genetic linkage between one genes to another genes. And then this is where the study of genetic mapping is occurs, right? So due to these particular studies, right? So some extra reading for you guys. Read the cytogenetic, genetic linkage, crossing over, and chromosome mapping. Try to find it in general, inshallah. Right? I guess that's all for today. All right. If you guys have any questions, do let me know. All right. I will share the slide in the iTaklim afterwards, after the class session. All right. Before we leave the session, do you guys have any questions?